Good morning. What I want to do today is to showcase, in the limited time I have, some of the interesting copy number projects which we have in our lab ongoing. I'll start with a project for renal carcinoma with rhabdoid differentiation. This work has just been accepted for publication in Human Pathology. And then I will go on and share with you some preliminary data on copy number analysis in samples where we have done concurrent NGS assays and FISH assays so that we could see which technology would help us in detecting copy number, especially in respect to targeted hybrid capture type of NGS assays. So what is rhabdoid differentiation and why do we care? If you look at morphology, for those who are not pathologists in the audience, these cells are with eccentric nuclei and they have eosinophilic cytoplasmic inclusions. And EM would show intermediate filaments and organelles. And rhabdoid differentiation is found not only in renal tumors but also in non-renal tumors such as bladder, stomach, and other organs. And it is a word of caution here, it should not be confused with the MRTK, malignant rhabdoid tumor of kidney, which is a separate entity which mainly occurs in pediatrics patients and has a well-known pathogenic alteration, which is biallelic inactivation of INI1 gene. And rhabdoid differentiation is found mostly in clear cell renal carcinoma, but also seen in a variety of others, but the majority is seen in clear cell. So other variety of renal tumors are chromophobe, papillary, cystic diseases, and medullary carcinoma, and also malignant mixed epithelial and stromal tumor of kidney. So it's seen in a wide spectrum of renal tumors. And so why do we care? I think the most important reason for this is that this is a very aggressive, clinically aggressive variant, and the tumors are usually categorized as higher grade, they have higher stage and a very bad prognosis. And there are no specific genetic alterations which are known for characterizing rhabdoid tumors. There are some of them which are published but not reproduced a lot. So there's a deletion of 3P and VHL mutation. There's a combined loss of BAP1. BAP1 is a gene which was found in uveal melanoma as well. So it seems more like a generic gene which is involved in other types of cancers. So, and then there are, based on the findings, we see that these patients respond to tyrosine kinase inhibitors such as sorafenib and sunitinib. So what we did is we used high-density SNP arrays. We started with cytoscan arrays because that was validated in our lab, but then we went on to use OncoScan arrays because they were much easier to use on FFPE samples and needed less amount of DNA, and lots of our samples were very old, from 5 to 15 years old, which we pulled out from our tumor bank. And then we compared with genome-wide alterations with or without. So within the same tumor lesion, we would have rhabdoid differentiation areas and non-rhabdoid differentiation areas. So it was a very nice control within the same tumor sample. So this is just to show you how rhabdoid differentiation and a clear cell looks like. This is rhabdoid and this is clear cell. And you can see, morphologically, morphologically, it is possible to differentiate these rhabdoid differentiation with the clear cells. So we took within the same tumors, as I said before, the rhabdoid areas and compared with the clear cells. So there were two separate arrays per sample which we ran. So this is just a clinical summary. The patients were from 42 to 73 years old. We made sure that the genders are equally represented. Majority of the patients were Caucasians, and this is the last bullet point, shows what the clinical outcome was. We were able to get clinical outcomes based on our consent, that about 50% of them had died, 40% of them are living, and 10% were lost. Two patients were lost to follow up. So this is how it looks like. You see that there are much more. It's very easy to just appreciate. This is all the patients together look, all the patients put together. So you see in rhabdoid cell areas, you can see that the copy number alterations are much more aggressive as compared to clear cell, and it becomes very obvious when we go and look at individual cases. So the slide which I showed previously was accumulated data, and this is a heat map showing the different regions, and I don't expect you to look at and appreciate the difference, but you can see the top panel shows how many copy number alterations are seen in individual cases as compared to the clear cell. So it is very obvious that the genetic alterations in rhabdoid differentiations are considered significant. In clear cell, what we saw that the gains were mostly seen in 5q and 7 and 8q, and losses were most commonly seen in 8p and chromosome 22. In rhabdoid differentiation, the gains were mostly seen on chromosome 7, and the losses were seen on 9, 14, 4, and 17p. So what we wanted to see is, how this kind of study can help us better diagnose these rhabdoid differentiation and, help the patients with aggressive tumor type. Overall, rhabdoid cells showed more genetic alterations compared to clear cell. And I've listed the major ones: 9 loss of whole chromosome 9, loss of 11q, and 17p. So now finally we have a tool which needs less amount of DNA and can tolerate degradation of DNA and can be used for helping our diagnosis here. So I've listed also some of these biallelic inactivations like we see in BAP1, PBR, M1, and VHL. Seen in both clear cell and rhabdoid. So as previously documented, these are not specific to rhabdoid. So we see that there are greater number of alterations, and just by looking at the number of alterations, one can use that to differentiate these rhabdoid and clear cell population within the tumor. And they are more genetically complex, we learned, and we saw that there were numerous chromosomal gains and losses. And overall genetic alterations in rhabdoid cells suggest that this evolves out of the clear cell, and it's a histomorphologic progression. So that was our conclusion of this paper, and as I said, it's available in Human Pathology this month. So I want to go on and show you some of the screenshots of the two different approaches we use, only a couple of cases to show how OncoScan was much more clearer and very useful in data analysis as compared to Cytoscan, which is a fantastic product on its own, but we use it mostly for inherited disorders in pediatric and prenatal situations. But for tumors, we see clearly that these two cases shown here have a very nice delineation of copy number as well as loss of heterozygosity areas, and I show here two separate examples: OncoScan on this side and Cytoscan on this side. So the data analysis is much easier using the OncoScan for FFPE samples. So in last 10 minutes, what I want to do is to share with you preliminary data of our evaluation of copy number alteration study and comparing with the NGS and the FISH data. This was done by Eric Dunkavich, Heli, and our colleagues from WashU. I had funding for this. So this is just to give you a background what NGS assay we use in our lab. We have a panel of about 50 genes. The genes are listed here, and we have been doing next generation sequencing for over 3 years now, and the subset of genes are responsible for heme malignancies as well as solid tumors, and there are obviously some common genes which are responsible or causative in both the tumor types. So this is how we design our NGS panel. We don't do hotspot analysis, but we look at the whole gene We look not only at the exons, but 200 base pair on the either side of the exons, and also we look at a KB on each side of the gene. So it's very comprehensive, and we look at all the whole coding exon and plus the intronic region, some of it. So this is overview, and I think I will be preaching to choir to show if I go over this assay, but we fragment the DNA. We fragment the DNA and we make a library using Agilent Sure Select method, and after the sequence is captured by Illumina HiSeq 2500, we align the data, and then depending on what we are looking for, we have several different bioinformatic tools which we use for SNV indel detection and translocation. And then we have our in-house developed bioinformatic software which then would put it all together for us in a very user-friendly browser, and then we generate the report which goes into the medical record. It's all good for single nucleotide variants, but for CNVs, it has been the NGS targeted based approach has not been very good, and here is the reason for that. We look at 1 kb to 1000 kb of sequence, and then the targeting panels depend on small fraction of actual CNV event. And sequencing of bulk genomic DNA makes it very difficult to detect CNV in small populations. We have to normalize the coverage, and the panels contains genes that are likely to be amplified or deleted. So if it's not a straightforward deployed genome, it is not very easy to detect copy number variation in a targeted NGS panel. So there are lots of different tools available. We have tested different bioinformatic tools shown here, Contra and Exome CNV and Copycat, which was developed at WashU by our lab. So we have been testing all the different copy number variation on the NGS data to see if we are missing any clinically relevant or actionable copy number changes. And as you know, just by the number of reads you can get, one can easily look at the major amplifications. And so shown here is an example of EGFR amplification, which you can see that if you look at the coverage, the coverage suddenly goes from 800 to 1,000 to 8,000, and you can see these obvious amplifications by NGS, but the smaller ones and the less copy number ones are not very easy. I show here we convert the data into log2, which can be used to detect the EGFR amplification, as I show here, but there are lots of other ones which are not very easy to be detected by NGS. So what we decided to do is to go back and look at our data, different tumor types, and also look at copy number alterations which we missed by our clinical assay, and compared it with NGS and FISH. So what I'm presenting here is early analysis. So I don't want to go through this whole list of all the 50 genes we have, but you can clearly see here there were lots of, so this is the OncoScan data, and we show the concordance between FISH and NGS here. So there are lots of these additional losses and gains which we have seen in our tumors, which were not detected both by NGS and FISH. Shown more here. So this is the summary of our concordance studies. So we could see the FISH detected amplifications, and it was not 100% concordance, and we think mostly it could be because of the low tumor burden of the sample and different sampling issue, but also we think that it is due to the fact that the NGS captures the total DNA and FISH is done on marked area based on the morphology. And we're further doing more studies with FISH probes specific to the ones which we saw in OncoScan to see. As far as NGS concordance is concerned, we see, as I showed before, the EGFR amplification is very easily detected, but the other ones are missed because of the fact that we don't have a total whole genome backbone which we can use to normalize. Specifically, we saw some concerning genes which were found by OncoScan arrays but were missed by NGS. We missed 4 homozygous PTEN deletions. And our assay, when we validated, was not meant to pick up these deletions. And now we have additional bioinformatic tools which we are validating, which can, hopefully help us. But loss of heterozygosity, all of them were not detected by our assay. Just wanted to share one case with you because I'm limited with my time. This is a kid with glioblastoma, grade 4, and had 50% tumor population based on morphology. And the NGS and FISH was done, so NGS showed EGFR amplification. FISH, of course, confirmed that, and was negative for MLL and ALK rearrangements. I show here the EGFR amplification by FISH. And as I showed before, NGS was very obvious because of the increase in the reads. OncoScan, however, showed many more additional clinically relevant data, multiple regions of LOH, EGFR amplification, which was also seen by NGS. In addition to that, there was CDKN2A homozygous deletion, which is actionable, chromosome 10 monosomy, P10 homozygous deletion, NBA, which is also has clinical actionability, and there were other chromosome gains and losses. And P10 homozygous deletion I show here was beautifully seen in using our software which we use in our lab. We use Nexus Biodiscovery as well as the CHAS software, which now can be used for OncoScan. And so I just show you how the 2-copy loss looks like in this case. So you have single copy loss clearly visible, and a 2-copy loss here, which is indicative of homozygous deletion in this P10 gene. So this is just a beginning. We are beginning to analyze more data and going back on the NGS. But I show here the frequency of these deletions, important deletions in all the 40 samples we tested. So you can see here there are key genes which are amplified here, EGFR, P10, and they are relatively high frequency. And I think the ones shown here are just the tip of iceberg, and these are the most important ones which are shown, and there are others too. So MET, EPH-A3, these are all clinically relevant genes. So I think until we have better tools, or and not only better tools, but the NGS, until it becomes the reads becomes longer, we will continue to have problems in CNV detection. I think OncoScan is a very useful tool to detect CNV in these tumors, and it's an important adjunct in our opinion. So we are obviously going back on the copy number NGS analysis. We're looking at this copycat tool, which was developed at WashU by Eric Dunkovich in our lab, in our group, and which detects the CNVs based on normalized log coverage ratios between sample and a pooled normal control. This is available on GitHub for anybody to download and look at. So, in conclusion, we definitely need better assays to look at copy number alterations in these solid tumors, which are today, because of the short reads and the problem of normalization with the normal genome backbone, it is very difficult. In the end, I would like to acknowledge my lab and especially the Dr. Humphrey, who just moved to Yale. He's now in Yale. Carmen, who's the resident. Vishwa is a clinical genomics fellow in my lab, and Mike Evanson. And these are the people who helped in the NGS study.